As the slide says, we're trying to deliver on the promise of iPSC-based T-cell therapy. And why is that? Next. There we are. So again, everyone, the promise of cell therapy as a whole, we're all intimately familiar with this. But despite many advances in the autologous cell therapy space, we're still, restricted to it's one patient at a time. There is variability, and accessibility remains a key constraint for certain patients. The former FDA commissioner, Scott Gottlieb, I think said it very well, is that today when we look at clinical trial results, when you aggregate data in autologous cell therapy, it's tough to know if the variability that we see in the trial results are due to the biology or due to the manufacturing process and the inherent use of different cells for every patient. So in the world of trying to create off-the-shelf T-cell therapy, where we really are trying to turn cell therapy from a procedure to a medicine to be treated just like an antibody or any other biologic, it's really having an unlimited supply of cells that can sit on the shelf and be ready to be dispensed to the patient. And what we believe that we've accomplished at Notch, and I'm going to show you some new data today, is that an unprecedented production of functional T cells from iPSCs. And I'll explain a little bit more about what the challenges our field has been facing. But it's also, by doing that, creating those cells using a proprietary manufacturing system that can be scalable right from your needs from a preclinical stage all the way up to commercialization without employing any new technology. So the first off is just a little bit about Notch. We are a company that now has 3 locations, about just under 90 employees. And we intentionally built the company to have the capabilities of having end-to-end capabilities for iPSC therapeutics. So we have our GMP and Process Development Center of Excellence in Toronto. Our good partners, the Center for Commercialization of Regenerative Medicine, it's in Toronto, and they were one of the co-founders of the company, as well as 2 of the scientific founders. Dr. Juan Carlos Aniga-Fluker, who is probably considered the grandfather of T-cell differentiation platforms, as well as Dr. Peter Zanstra, who up until recently acted as our CSO and is well regarded and well known in the whole bioengineering field of scalable systems for cell therapy. In Vancouver, we have our GNA facility, our team, as well as our stem cell biology research team. And then in Seattle, it's really what I'll loosely call our product development team, though today it's really focused on protein and genome engineering and translational sciences. In the future, that's where we would also expect to be building our clinical and regulatory infrastructure. We've had some very important milestones along the way, including a partnership with Allogene Therapeutics, where David Chang, as the CEO, really sees the vision of where cell therapy needs to go, and that an iPSC-based approach will enable that. And then as well, a world-class group of investors backed us in early 2021 to really advance this company. So this is not unique to Notch. Everyone in the iPSC field has the same vision of how to go from a pluripotent stem cell that could have theoretically unlimited cells from one donor, where you could do multi-gene edits to make that into a highly potent medicine, to then be able to have a universal master cell bank that, as needed, that becomes your starting material to produce scaled generation of high-quality cells that can be sitting on the shelf for patients. But the reality is, and where we have focused and differentiated ourselves as a company in this space, is that the actual production of scaled, high-quality iPSC T cells has not been as successful as we would like to have been. And our whole focus has been, rather than focusing on the gene editing part at this point as a company, we think there's lots of companies doing great gene editing, have great receptors, great other tools. If you don't have a good cell, all of that gene editing can go to waste. The highly scientific term of garbage in, garbage out. That if you're putting all of those edits into a very weak cell, that you may not end up with a highly potent functional cell that can do the things that a primary T cell can do. And you may have seen it, but if you didn't, I encourage you to look it up. But we recently published in Nature Biotechnology a paper about how our systems biology team deconvoluted clinical data in the autologous cell therapy space. And no surprise that, that modeling really reinforces that cells that can proliferate when exposed to antigen and persist are critical for clinical response. And this has been the driving force behind our process of how to get those cells that replicate what a primary T cell does. Now, we are not saying that CD8 alpha beta cells are the only interesting cell in cell therapy. We think all of the work that's being done on gamma delta, NK cells, and others is all very interesting. But as a company like Notch needing to be focused, we see that alpha beta T cells is where the clinical precedent is. That's from where we've learned everything about cell therapy in the clinic today, from approved products like Kymriah and Yescarta and others. And so our goal has really been trying to replicate in a synthetic way what we see from those primary CD8 cells. So in a moment, I'm just going to show you a few little movies, but at first we just want to explain across the top what we believe is unprecedented data. So this is where we compare our iPSC CD8 CAR-positive cells. So the target is not disclosed here, but it's a CAR-positive cell, showing that it can, act through multiple rounds of in vitro stimulation against the cancer cells and effectively kill those cells, as well as a primary T cell that has been transduced with the same CAR. On the far right-hand side is really where we think we have really differentiated ourselves, is not only are we making cells that phenotypically look a lot like primary T cells. I'll never say they're identical, but they are very, very close. But they proliferate in response to antigen, just like a primary T cell. And we think this is going to be critical as we go to the next step for in vivo, because we think that proliferation in response to presentation to antigen is what leads to persistence in vivo. And I just showed two references there. Typically what we're seeing in the literature from other approaches, other cells that have been made from iPSCs, you're typically seeing under 100-fold proliferation. And so we're starting to see the levels of proliferation in response to antigen, just like you would with primary T cells. So to see that in moving pictures, if we could just show the first video. So this is, again, when you have no CAR-positive cells present. You just want to click on that. You see the fluorescent green is the proliferation of the malignant cells in culture. Then you go to the second one. You'll see as the T cells with the CAR are present, they will just start to destroy and kill the present cells. And then as far as the fourth stimulation, the same level of potency and ability to kill those cancer cells. So I think I can sit here fairly confidently and say we have not seen data like this anywhere in the literature. And I'll be showing you a slide in a moment about some of our upcoming presentations, because this is the first year that we're starting to show this data. So I think we feel that we have accomplished at scale the kinds of things that we need to have what we believe is what we've all been searching for in the iPSC field, where you have a highly uniform product with reduced variability on batch-to-batch manufacturing, having all of the right characteristics to get us to the proliferation and potency. And when I talk about scalability, it's really because of our approach. Our inherent technology is how we control Notch signaling through the use of magnetic beads in a stirred tank reactor. And that's what we call our engineered thymic niche technology. And it's the ability is to not only now present the right signals to the pluripotent stem cells to have them differentiate the way you want, but we can control that signal. We can control the density of the signal, the duration of the signal, but also being in a stirred tank reactor, we can control oxygenation, feeding, all of the other things that are important to be able to have healthy cells at the end of high consistency in terms of phenotype, as well as in terms of their viability, as well as reduced batch-to-batch variability. And the beauty of what we're doing is we're only doing small-scale experiments now, so we can do this in 250 mil stirred tank reactors, but we've already done proof of concept to show that going to 1 liter and 3 liters, there's no change in technology. We get similar yields. And so as we start thinking about clinical trials, we believe that probably somewhere around a 5 to 10 liter batch is all that's going to be required to produce over 1 trillion cells that would be sufficient materials for 2,000 doses. So we think that this is very much game-changing in terms of how we think about small footprint manufacturing to be able to have something that's readily scalable, less expensive, dramatic reduction in cost of goods. If you speak to people at Kite, they will tell you that it's almost a 1-to-1 ratio between operators required for every dose produced. So as you go up in number of doses produced, you need more and more FTEs. For us, the same number of operators can produce any one of these bioreactors. And so what we'll hope that this will eventually translate to, and I appreciated the comments of the last speaker, it's all about the patients. How do we get a product that's more accessible to patients with reduced variability, so that should have better clinical results and reduced wait times? Of course, we have not forgotten the need or the desire to have cell engineering, so we've also incorporated that in our business and are now starting to work with partners about how to put in the right targets, the right other switches. But we really believe that fundamentally starting with a great cell and now doing these edits is what's going to translate into a highly effective clinical product. So in our roadmap to a successful product, we start with a commercially ready iPSC product, which we reprogrammed ourselves under GMP. We have our own allogeneic chassis. We have a systems biology approach that guides the exact phenotype that we want so that we know that we'll get that proliferation to antigen that we think will translate into great persistence without engineering. If you add things on top of it that you engineer in, we think we could even get potentially even better persistence. But starting already with a cell that we think will act naturally like a primary T cell. The function is standing up against anything we've seen, and it's using this bioreactor-based manufacturing that we'll be able to accomplish, again, everything from preclinical to commercial scale. So where we are today is we already have our partnership with Allogene, and now we're in discussions with others for other CARs or TCRs, or anybody just that has another idea that they would like to see in functional T cells. So if any one of you are in that category, I would love to spend time with you. So as I mentioned, some of this data has been presented this year. There's a number of publications that are available on our website, the 3 bullets at the top, about how we've been able to control the cell phenotype using that engineered thymic niche. Probably one of the things that I think is being underutilized within Notch is our ability to produce massive amounts of CD34 cells, again, using bioreactors, high purity, that have very strong, again, similarities to hematic progenitor cells based upon RNA-seq phenotyping. And we're using that as a starting material in our differentiation to T cells. But if there are people that are interested in CD34 cells, we pretty much have an unlimited supply and would be interested in talking to you about how that could be a starting material in your process. What I draw your attention to is some very important upcoming presentations where the scientists will be going into much greater detail about what I talked about today. So we've already been accepted for presentations at ASGCT, ISCT, and ISSCR. And so I think you're going to be hearing a lot more about Notch in the coming months and years. I referenced the paper that just came out that I think, again, speaks to the quality of science we're trying to undertake at Notch. And it really comes from having, I believe, one of the strongest teams in this business, that bright minds come up with innovative ideas, and there's a lot of experience on the team. And it's always been with this fundamental focus of how do we turn cell therapy into a drug so that we can make it accessible to many more patients. So I thank you very much for your time, And I don't know if there's probably no time for questions, but if anybody wants to speak, I can be waiting outside. So thank you very much.